Hello, welcome to our second video on nutrition in plants. Now I'm just going to start off this video by saying that please go and read your textbook, page 127 for Pure Bio, on why is photosynthesis important. This is a question that does sometimes come out in O levels, and there are three big reasons why it's important. Please go and make yourself familiar with them, memorize them, know how to describe and explain them. The second thing is, there's actually an optional video for this lesson, for this set of notes, which has more details on photosynthesis, but it's really optional. So only watch it if you want to relearn more about photosynthesis or have a deeper understanding of it. So let's start with a quick recap on, hopefully you can still remember, what are the raw materials of photosynthesis, the products, and the conditions. So pause and try that for a while. So the raw materials are carbon dioxide and water, right? The products, glucose and oxygen. And the conditions, light and chlorophyll. And we can put those together into the equation. And again, the two important equations that you need to know, the word equation and also the chemical symbol equation. Right, both are important and you need to know it well. You need to memorize it. Well, I'm just going to spend a bit more time on this. So you can see now that this is carbon dioxide, water. This is the chemical symbol for glucose and oxygen. And we have, this is to show the ratio. Right, so it's really 6 carbon dioxide molecules and 6 water molecules become 1 molecule of glucose and 6 molecules of oxygen. And that's what this equation means. So in this video, we're going to be looking at how can we study photosynthesis. Now look at this little girl here looking at the beautiful leaf, studying it. And she's thinking to herself, how can I tell if photosynthesis has taken place in this leaf? Well, after all, it's a good question, right? The leaf is just there and it's not doing, doesn't look like it's doing much. It's kind of leafing around. How can I tell if photosynthesis has actually happened in the leaf? So we're going to be looking at some of the experiments that people do to study photosynthesis. And like this guy here, you just look at the plant and say, I think we just saw it do photosynthesis, Mr. Grant. But how do we know? So in this video, we're looking at how do we study photosynthesis. We'll talk a bit about this thing called destarching, which is done in a lot of experiments with photosynthesis. And more specifically, how do we study the conditions like light and chlorophyll that are needed for photosynthesis? These are the textbook pages for pure biology. So in this video, we're going to be looking at a lot of quirky experiments. And there are a lot of these kind of questions that come out in the O levels. So it's important for us to also know what these experiments are looking at and why are they designed a certain way. So how do we know if photosynthesis have taken place? Well, let's look at the word equation first, and we can get some ideas from there. The word equation tells us what is used up and what is produced during photosynthesis. So one of the ways that we can study photosynthesis might be to measure the volume of carbon dioxide being used up. Right, as carbon dioxide is used up, I know that photosynthesis is taking place. Now there is a way of measuring carbon dioxide being used up, and that's through using an indicator called hydrogen carbonate indicator. This indicator basically changes color as carbon dioxide is being used up. Okay, go read up more about it in your own time. I'm not going to be talking about it much more. Now, of course, a second way that we can study photosynthesis might be to measure the volume of oxygen being produced. As oxygen is being produced, we know that photosynthesis is taking place. Now, this is slightly more common. So, for example, I put some underwater plants, like this pondweed, I immerse it in water, and I give it some light, and I see that after a while there are bubbles being produced. Now these are in fact bubbles of oxygen gas. Oxygen is being produced during photosynthesis, and that's why they are bubbles. So as I kind of count the number of bubbles, I know that photosynthesis is taking place, and I can also measure the rate of photosynthesis. But how do we know that the gas produced is oxygen? Alright, let's watch this video to find out. So in this video, you have seen that this guy has collected some gas, right, oxygen gas in the test tube above the plant. And what he's going to do, see he's collected it here, he's taken out the test tube, is he's going to light up a splint. And it's going to be a glowing splint, and he's going to insert the glowing splint into the test tube. So watch. Right, and then if you saw that, I'll show you again. He's inserting the glowing splint.
Okay, so that's letting in a tiny bit of normal air. And then blow that out. Hey!
Alright, so lots of happiness there. Well, basically, we can test for oxygen because oxygen will relight a glowing splint. So we can actually put a glowing splint into a container containing oxygen, and if it relights, then it tells us that it is oxygen gas. So oxygen gas relights a glowing splint. Cool. So we've seen that we can know that photosynthesis is taking place when we see oxygen being produced. Now there's one more way that is often used, and that is we know that photosynthesis has taken place if we measure the amount of glucose being produced. Now you don't measure glucose directly, usually people will measure the mass of starch produced. So as starch is produced, then we know that photosynthesis is taking place. So in many of these experiments where we measure the amount of starch produced, how the experiment is done is that we start with a destarched plant. Now a destarched plant is a plant with no starch, right? The leaves have no more starch, and I'll talk about that in just a while. Now this destarched plant, we carry out an experiment. So for example, we put it in the light for 24 hours. And then at the end of the experiment, we test the leaves for starch. Now if starch is present in the leaves, then we know that photosynthesis has taken place, right? Because previously, the leaves had no starch. It was destarched. Then we put it in the light for a while, photosynthesis happens, and then the leaves have starch. So if there's starch, that means photosynthesis has taken place. So what is destarching and why is it important? Well, how do we destarch a plant? We put it in the dark for 48 hours. So the plant will use up all the starch in its leaves and will leave the plant with no starch in its leaves. Now the importance of this is this. Destarching ensures that starch is absent, that there's no starch in the leaves prior to the start of the experiment. So at the start of the experiment, we make sure there's no starch in the leaves. And this is to ensure that any starch detected at the end of the experiment was actually formed during the experiment. That means the starch wasn't originally there, right? The leaves had no starch at the start. Then at the end of the experiment, any starch that is detected was produced during the experiment. So how do we test the leaves for starch? Well, you're probably already telling me that it's something to do with iodine. Well, there are actually a few more steps involved, and we'll try this in the lab next time. So I think let's skip over this part first, but you can actually find it in your textbook as well if you want to have a head start. So there is a table there, and you can fill it up. I'll put the video up on the tiny URL as well, but we'll do this in the lab next time as well. But first, we're going to end off by looking at some experiments about photosynthesis. And we need to remember what these three terms mean: independent variable, dependent variable, and control variable. Remember this experiment where we did the potatoes in different concentration of salt solution. In this case, remember that the independent variable is the one that I am interested in, is the one that I change. So that is the concentration of salt solution, right? The dependent variable is the one that you measure, right? The one that depends on the independent variable. So this one is the change in length. And of course, the control variable is everything else that you want to keep constant, like the temperature, the type of potato, right, to ensure a fair experiment. So I'm going to use some of these terms in the next few slides. So let's go. So this is an experiment to investigate whether light is necessary for photosynthesis. Right, you see a leaf here, and basically the leaf is covered with some black paper, right, on some parts of it, but there's a cutout here, a star-shaped cutout. So this part, the leaf is still exposed inside. Now in this case, what is the independent variable here? Dependent variable, control variable, and expected result. Like where will there be starch at the end of the day? I want you to just think about it and then pause the video and I'll talk about it in a while. Well, the independent variable is light, right? Some parts of the leaf here have light, while other parts that are covered by the black paper have no light. So that's the thing that I'm changing. What's the dependent variable? Is the amount of starch produced, right? At the end of the day, we measure whether there's starch in the leaf or which parts of the leaf have starch. And that also is the amount of photosynthesis, right, that produces that starch. That's the dependent variable. Okay, control variables be things like temperature, where do we put the leaf, what kind of leaf, and what's the expected result? Well, you would expect the parts of the leaf in the light, this part would have starch, right? The parts covered have no starch because no photosynthesis happens. And the part here in the middle with the cutout, there'll be starch there too. Right? So this experiment shows us that light is necessary for photosynthesis. Now this is a second experiment. It's about whether carbon dioxide is necessary. There's a potassium hydroxide crystals in one setup. Now these crystals, they actually absorb carbon dioxide. So if you put these crystals here, they'll remove all the carbon dioxide from inside this bell jar. Alright, so here is a bell jar without potassium hydroxide crystals. So here, there is carbon dioxide inside. Once again, think about what are these variables in this experiment. Pause the video and I'll give some of the answers. Now in this case, the independent variable is carbon dioxide. Right, in this setup, there's no carbon dioxide because it's absorbed. And in this setup, there is carbon dioxide because I'm interested in knowing whether carbon dioxide is needed for photosynthesis. Now the dependent variable, again, is the amount of starch in the leaves at the end of the day, or the amount of photosynthesis that has taken place. The control variables will be things like, where do I put these two pots? I must put them in the same kind of light intensity. Or, what kind of plants are these? They must be the same type of plant. And the expected results would be that this plant here has starch, right? Because photosynthesis took place. But this plant here would have no starch produced because no photosynthesis took place. And that's how we know that carbon dioxide is necessary for photosynthesis. Finally, this experiment here is to investigate whether chlorophyll is necessary for photosynthesis. For this, we use what we call a variegated leaf. Now, a variegated leaf is a bit like a money plant leaf, right? Some parts of the leaf are green. So here, this part is green, has chlorophyll. Some parts of the leaf are white. They don't have chlorophyll, right? Then we put this leaf in the sun and we test it for starch after a while. Now think again about what the variables are here and the expected results. Now in this case, the independent variable is the amount of chlorophyll, right? So some parts of the leaf, like this green part, have chlorophyll and the white parts don't have chlorophyll because we're interested in whether chlorophyll is needed for photosynthesis. The dependent variable is the amount of starch produced and the amount of photosynthesis that takes place. The control variable will be things again like, where do we put this leaf? Make sure that all parts of it have equal amount of light, blah, blah. And the expected results is that the green parts of the leaf would have starch, right? Because they have chlorophyll, but the white parts would not have starch because they have no chlorophyll. Alright, that's the end. one last video before you end.